Okay, this is the talk that I was going to give before Barry said do the one on Doctor Who, and so I decided we have a spare slot, I'm going to do this anyway. As you know, I'm lucky enough to work on Scala, very modern operating system, very modern language. I love my job, but it's not all roses. And I'm going to talk today about the legacy system that I work with, which is the most horrendously complex legacy system that you can think about. It's in production everywhere. It's constantly being forked. There's major releases only every 20 years or so, but there's these patches that happen all the time. Literally, it's being patched all the time. We don't have the source for this thing. We've only got the binary for it. Much of the code apparently does nothing at all. It's hard to tell what is and isn't important looking at the code. DRY is violated everywhere. There's copy and paste all over the place. And much of the code, a huge amount of it, is actually infected by viruses. The binary is about 3 gigs. We have a handle on about 100 megs of it at this point. The rest may be junk, or it could be critical to operation. We simply don't know. And any given deployment can be tricky or even dangerous. And just so you know, this is the bit that we think we know about. This bit is about a bunch of metadata that is important. We know it is important, but we don't know what the hell it does. Down here is a whole bunch of kind of cut and paste code that we can use, and it's there. It's a resource for us. This is duplicate data and just basically garbage. And this is the stuff that's infected by viruses. Okay? Pretty bad set of things. Our tools that we've got grep and cat to use. The greps are incredibly slow. The regexes are extremely long and complicated. And you can make some guesses at the outcomes of a run, but mostly it's observation. So, do you want to see the code? Anybody guessed yet? Okay, here's the code, and here's a new release. So anyway, you probably guessed I'm talking about human DNA. We've got the tools that we've got to work with. We've got a basic disassembler at this point. It's called protein folding. It's about 80% accurate, the prediction rate for it. We've got a thing that's like a profiler. This is the kind of the regexes, the grep. There's various tools. The leading one is fluorescence. It's a way of detecting what DNA patterns are available. The debugger, basically, we're not going to see this anytime soon. We might see it in our lifetime, the ability to actually see the code as it's running. That would be a wonderful thing. The testing framework is a bad story. Okay, it has 0.0% coverage. All testing is carried out in a production environment. There is literally next to no documentation. Less than half of the 25,000 genes even have an overview. Almost none of them have any kind of detailed documentation at this point. Backup copies is actually a good thing. Every cell in the body has a full copy. So in every body there's 10 trillion backups of the code. The only problem is the backups are flawed. They vary in each case. There's very limited off-site backup, and there's no restore, at least not legal. There is a caveat here. Believe it or not, sportsmen or sports people actually use gene doping now. They inject DNA using retroviruses to enhance their performance. It's incredibly dangerous, but not recommended at all. There's corruption in all the copies. There's no verification. There is some basic error correction, but it's not flawless. Basically, each bit may only be in 2 of the 4 possible states. Corrections are a guess. It's better than nothing. It catches some stuff, but it's not. It's certainly not perfect. Other error correction mechanisms tend to be a little bit more final. Natural selection is kind of the ultimate error correction. Forks, about 1 in 1,000 is different in 2 random human beings. We know about the ones in purple. The others from the earlier slide are basically undetected. We talk about this thing called SNPs, which are single nucleotide polymorphisms. These are gentler changes. They're like bit rot in code. Occasionally you get wins from them. The other thing, though, that's really interesting, you remember that a third of the code that I showed you that was virus infected? They're called transposons, and they are responsible for the large jumps forward in evolution that let us deal with catastrophic events or life-threatening changes in our environment. What happens is the cells go to this area of virus infection and actually pick out code, and usually it's trash and it doesn't work. Occasionally it can lead to stuff. And conclusion, very, very quickly, in spite of all these problems, this is an incredibly successful system. And maybe we don't know as much as we think we know about deploying really complex systems. So there you go.